Thank you very much to all the organizers for this opportunity, this invitation. I'm really happy to talk to ICTS online and to the community in stochastic thermodynamics. This was my promised title, and I'm really sorry I'm not going to be able to talk about this. But the reason is we are writing a review that is coming soon. It's called Martingales for Physicists. It's a tutorial review in which we give our viewpoint on stochastic thermodynamics with martingales throughout 3 very extensive chapters, which you will be able to read hopefully end of summer. In particular, this chapter 6, which you will see, contains the so-called football club martingales of second laws. Here we showed how to derive most of the well-known expressions, formulations of the second law from just the martingale property. I'm not going to give details, but this is also my opportunity to ask if someone is interested. We are looking for people who are ready to give us feedback before we submit the review. All right, so I changed a lot of gears, and I will talk about active fluctuations in the ear of the bullfrog, which is more related to what Simone and Mike just explained. In particular, in my group, we are highly interested in sound transduction in Bullfrogs Sacculus, and also as a general problem, in highly specialized cells, which are called the Eair-hair cell bundle, which are responsible of transforming the sound into an electrical signal by using a chemical energy, because these Eair bundles are powered by molecular motors, as I will explain later, which most of which the chemical energy is dissipated into the thermal bath as heat. So the teaser of my talk is the following. We would like to estimate the minimal dissipation or heat dissipation required to sustain a single active oscillation that can be measured by tracking the tip position of this Eair cell. This is the teaser, and the talk will be mostly about this. So why bullfrogs, and why we do really care about this? There are many reasons. It has an unusual, important biological function, of course. But it's also interesting because only 2 degrees of freedom are sufficient to describe the dynamics of such a system. There is 30 years of stochastic modeling, so it's as much as the round tumble. They are stereotype and highly reproducible, so you can do experiments and control conjugate variables, such as the displacement of the cell, but also the current that the cell is generating due to its motion. And also that there are many common structural and functional features across species. So you can see here the microscope image for the hair bundle in bullfrogs, pigs, and turtles. They are very, very similar. Another important thing is that ear herself will not regenerate. So when you have a damage in your ear, it's over. So this is also a big reason why we would like to understand how they function and how they operate. So first, I will have to dress up as a biologist for a short time. So, okay, very briefly, the ear works as a microphone. So you speak to the microphone, and the sound, the mechanical vibration, is transformed into an electrical signal. And this is the main function of the ear through its different components, especially the inner ear, which is made of this snail-like cochlea, which I highlight here, which is has this spiral-like structure in which the membrane, the vascular membrane of the cochlea, is endowed by this set of cells, which are called, these are highly specialized cells, which are called Eair cell bundles, which are responsible of transducing sound into an electrical signal. This is a very complex process in which, that's another thing, is that these Eair bundles are placed in the membrane of the cells. And you see the cell is huge compared to the Eair bundle, which has a height of around 7 microns. And the function, it's a bit complex, but I'll try to summarize in a simple way. So the Eair cell bundles are formed of these filaments called sterocilia, which are connected by other thinner filaments called the tip links. And when the Eair bundle is displaced due to the arrival of sound, these tip links are stretched, and they open. This stretching makes that the tip links, which are connected to ion channels, open these ion channels and let ions to flow from the extracellular medium to the intercellular medium. So this is a way of mechanotransduction of the sound in a very brief explanation. But of course, there are many models that I will show later that explain this type of motion. On top of that, this is not just passive elements and springs, but there are molecular motors that are responsible of adaptation. So when the sound enters in the ear, you have a very loud sound, the ear doesn't break. So there are active elements that are able to do feedback and to adapt to a mechanical stimuli and to move dynamically these channels. This is done by myosin motors, which consume ATP, and these are some of those who carry the most of the heat dissipation in the motion of the hair bundle. Okay, so there are 4 unique features of this model, of this biological system. First is that they can amplify sound. So if you do an experiment and you move the cell by an amplitude of 10 nanometers, they can respond to the stimuli by amplifying the motion. And this motion is, there's frequency selectivity. Depending on the frequency, you will have a higher response or lower. And they are very specialized, so they can respond selectively to very specific frequencies. Moreover, in terms of the amplitude of this stimulation, they have a nonlinear response. And finally, what is most exciting for me is that they can oscillate spontaneously. So even if you don't stimulate, so if you don't move the base of the cell, they can still move as a function of time and display oscillations. So this has fascinated many scientists for many years, and we are trying to understand nowadays the thermodynamics of these spontaneous oscillations. To tackle these 4 unique features, there are many models in the literature. This is a very popular one, which we recently simplified to a 2-variable model. I will give details later on what are these variables. So one is the top of the cell, and the XA is the position of the center of mass of the motors in a way. This model reproduces the sharp spectra of X and also has a very interesting phase diagram in which experimental data has been shown in the past to be close to a Hopf bifurcation, which is a point in which if you move a little bit out of it, you are in a situation where there is a stable fixed point. But if you change the parameter, in this case, is the force of the motors a little bit, you enter in a situation where there is an attractive limit cycle, which is supposed, the theory said, can reproduce most of this phenomenology. So I will focus on these oscillations, and in particular, I report here one example of a oscillation from a bullfrog. Remember, this is activity, but this is post-mortem activity. So one cannot put a fiber in the ear of bullfrog while the bullfrog is alive. So one has to kill the bullfrog first, extract the cell, put it in a medium, and then after the frog is dead, this cell is active. So it's still displaying activity, or is doing this type of bistable oscillations, mainly because you try to reproduce physiological conditions in a medium where there is ATP. So you leave ATP and calcium in the medium, so the cell can use chemical energy to display these oscillations. This is from 2001, so there has been a lot of research, and the main first contribution in thermodynamics was this paper, which tried to compare the spontaneous fluctuations. So when you put a fiber on top of the cell and you see how it moves, with the response. So you stimulate the fiber with a sinusoidal forcing, and you see how this tip of the cell responds. And you see active matter is been there for a while, and this is a biological system where they could conclude it's really active. So we know from equilibrium physics, since Kubo, about the fluctuation-dissipation theorem. So if you have a system in equilibrium and you perturb it weakly, the response to perturbation is related to the autocorrelation function or the power spectrum in equilibrium, and via this relation, where T is the temperature of the thermal bath. So we may wonder if this holds in a hair bundle. So we can compute this experimentally, try to see if the ratio between these two is equal to 1, because this is a way of measuring the degree of violation of FDT. This will be equal to 1 if the previous equation holds. So they did this 20 years ago and found that if you put the cell with a channel blocker, which means it cannot display pure active oscillations, you have very clearly fluctuation-dissipation theorem. Whereas in an alive living cell, fluctuation-dissipation theorem clearly doesn't work, and you can conclude this is a non-equilibrium. So the reference process is a non-equilibrium stationary state instead of equilibrium. So in this way, you can say this is not equilibrium, but yet we would like to go further and say how far from equilibrium is this system. For this, we apply in a recently published work, but it's been historical. We worked for many years on this. We apply concept from stochastic thermodynamics. So for instance, imagine you have a stochastic system with two variables, and you can measure trajectories and time reversal of these trajectories. It's well known that the Kullback-Leibler divergence between the probability to see a trajectory and the probability to see a time reversal trajectory, it's a lower bound, provides a lower bound to the actual entropy production. And moreover, this bound is different if you have access to different amount of degrees of freedom. For example, if here I don't see x2 and I only see x1, I will have here k equals 1, and you see, it could be even that this number is very small compared to the real entropy production. So we really did a very systematic study on how this quantity, by changing the number of observable degrees of freedom, bounds the entropy production. This is what basically this inequality means. The more degrees of freedom you have, the tighter you get to the entropy production. For example, if you simulate these two variable model of the Eair bundle, you have these trajectories, and you compute the Kullback-Leibler divergence between forward and backward, you get around 1000 kbts per second, which is close to the total entropy production rate. Whereas if you cannot see the two variables, you only see one of them, you get, with some techniques that we develop in our work, much less entropy production, or much less irreversibility. So it's hard to conclude this is an active system. And this is mostly what happens in many of the experiments. In experiments, typically you don't have access to a second variable. So we developed a technique that I will not explain today, but if you have questions, I'm happy to explain it briefly later, which measures the time irreversibility of a one-dimensional signal, and we apply to experimental time series and show that one can see with scaling with the length of the trajectory that this quantity can predict up to 10 kbs per second in oscillations, which is, as you will see later, is nothing. And yet, even though this is small, one can discriminate between this situation and the situation where you have a zero, which comes, for example, from a reversible two-state model of Kramer's transition like this one. So still, it's useful to say, I am active, but the bound is not very tight. And this you can see explicitly when you go a bit forward and you say, let me do a simulation of the model. This is a published model on the Eair-hair bundle, where x1 is the tip position of the cell and x2 is the center of mass of the position of the motors. And let me compute for this model, which is simple Langevin dynamics with two variables, the real entropy production, which is just the Kullback-Leibler of trajectories with two variables. And in this case, it's very simple. It's the sum of two terms. One is the power from the first force. This is the F1 here. And the other is the power exerted by the second force on the second variable. Of course, if you want to do this analytically and go further, this is quite hard because generally this potential is nonlinear to generate these oscillations and also this force. So this is up to the place where we arrived. So we can come here and evaluate this in simulations. There's another strategy with two variables that concerns uncertainty relations. So you can, for example, construct a variable P0, which is somehow the transduction current. This is a variable that is depending on x1 and x2. You plot the transduction current versus the position of the Eair bundle x1. This is something that experimentalists will be able to do in the future. And you observe that in one side of the Hopf bifurcation, you have very clear currents in this phase space, whereas on the other side, there are not, observable currents, but yet you can do this analysis of computing the cumulated, for example, clockwise current, average square divided by its variance at finite time. this is a result, or technique that was highlighted in these papers. And of course, you can also do something more advanced, like what Isaac explained on the first day. But as for back then, when we did our work, we focus on this quantity. So what I'm showing here is a comparison of the different estimates of irreversibility as a function of one of the parameters that controls going from monostable to oscillations. So here we're crossing a bifurcation. Here I'm plotting the total entropy production in black, and the different colors are the different ways of measuring irreversibility. Yellow and red are with two variables, so you can do the thermodynamic standard relation for the entropy or the thermodynamic standard relation for the clockwise current, and you see that they are quite decent. So you can predict the almost 10 to the 3 kbs per second, whereas with one variable it's very limited. So you can go up to 10 to the 0 kbs per second, which is really 3 orders of magnitude below the real value. So besides giving this number, I think it's very important to connect this to biology and to compute entropy production in the most common units, the ones that make more sense. In my opinion, one has to renormalize this by how long it lasts a cycle. So how much energy you need to perform a single cycle of oscillation. In this case, the cycle takes 0.1 second roughly. So this accounts for more or less 100 kVts per second, which is roughly 10 ATPs, ATP hydrolysis per cycle. So it's really, this number is really cannot be obtained from a passive oscillation. So you really need 10 ATPs per cycle. It's more than a thermal fluctuation. So the 1 kVt per cycle would be really passive, whereas if you need more than 1 kVt, it means you need more than a fluctuation to perform all these cycles. So we conclude clearly the system is active. Very recently, with Gennaro Tucci, a PhD student of mine, now in Max Planck in Göttingen, we developed an alternative way of inferring the entropy production, which is by fitting the data to a very simplistic model, which is one variable model that is linear. So it depends, x dot depends on x. It has noise, thermal noise, but it is non-Markovian because it is driven by a dichotomous noise that can be non-Markovian. The model works like this. You have a particle in this blue potential diffusing as a function of time, and all of a sudden this potential is switched to the other orange potential, and so on. So basically what you're doing is you're switching the center of the potential at random times. This makes that the jumps upwards and downwards are not related typically to the difference of energy between in the jump. So this makes that the particle exhibits broken detail balance. Notably, we could develop our theory for generic waiting time distributions for the process CT. So if you pick, for example, exponential waiting times, you obtain for the signal X a Lorentzian spectrum. However, it's much more interesting to pick non-exponential waiting times, such as gamma. And there, if you do that, well, the dynamics is not Markovian, it's hard to solve, but yet it's interesting because the spectrum develops a peak like we see in experiments. So this would be more realistic to model the band. Actually, from experimental data from Jim Hasper's lab, we see that experimental data fits really, really well to a gamma distribution waiting times, which we use later to do predictions on thermodynamics. So here I introduce you to Alfred. So Alfred is one example of a bullfrog from where we took a cell and we did an experiment. You see here, these are the oscillations, and fitting the waiting time distributions, we can then do a simulation and see that the simulation in the computer is very similar to the experiment in Alfred. Also, the bimodal distribution and the correlation function looks an excellent fit. And by the way, let me just remark that we can solve that correlation function for generic waiting time distributions. We can solve, quotation marks, because we can do it in the Laplace space, which is already a lot of progress for us. And you see the power spectrum fits perfectly, but this is not the only animal where we could do it. Also here, Manfred is another example, and Lothar is another example where we can do this process of fitting and extracting the relevant parameters. So once you have the parameters, what you can do is try to estimate the power dissipated following Sekimoto. So we have a model where we can, using stochastic energetics, realize that there is work done only in the switches. All the rest, the particle is relaxing and dissipating heat. So there is only work when you act externally and change the parabola from one side to another. This makes the thing simple and let us compute analytically the work done on the system.
Arnab, what is C? C, can you just repeat that? What is C here?
Okay, C is the center of the trap. Okay.
Okay.
So in the model, the center of the trap is a dichotomous noise. It has two possible values and it's jumping with a waiting time distribution given by this.
Okay.
Okay. So you only have, you only do work when you change the potential, which is when C makes a jump. Okay, that's why you have this equation.
Okay. So these are sudden jumps, right?
In C, it's sudden jumps. Yes, it goes from minus T0 to plus T0 instantaneously. Does this solve your question?
Yeah, thanks.
Okay. All right. So for this model, we can compute the work. So this is the power is this amount divided by dt, average over many realizations, and it depends on the switching time, but also on the Laplace transform of the waiting time distributions evaluated at a characteristic time. And from the fitting, you get these parameters: nu, d, this is the diffusion coefficient, the stiffness of the trap, and we just plug it in the formula and get that for Alfred, Manfred, and Lothar, we get these values of, again, 10 ATP per cycle. So it's maybe a simpler approach to what I explained before. It requires only knowing one variable because we learned the C variable for the fitting, and it gives us the same number. And it also coincides with a recent work applying the Harada-Sasa equality. So it's really consistent and makes sense. Of course, this doesn't work for all bullfrogs. So some bullfrogs look bistable and the fitting is really good. But here, my friend Agustin, for example, has a different shape. You see it has like a shark fin shape. So yet we don't have a universal model that can fit also this type of shapes. So far, we can only fit those that look bistable. But yet, for this type of signals, one can use more generic approaches, which are based on measurement of asymmetries in waiting times, such as this work by Martinez, Gisker, Horowitz, Parrondo. There's also this very nice paper which finds a way of estimating the hidden entropy production in a two-state model by some sort of optimization criterion. And it was applied, for example, to cows sitting and standing up. And they conclude that cows consume at least 2.4 10 to the minus 21 calories per hour in deciding whether to lie or stand. So, of course, this is just on the process of deciding. But anyway, it's a really, really lower bound. So I would be careful on applying this to microscopic systems. We also have a different approach. And in the case where you can only observe few visible transitions, you cannot see states, and this is related to our work, but also what Udo Seifert explained on the first day. I don't know if I have more time. Do we have 5 more minutes?
Yeah, you have.
Very good. So I have 5 more minutes to briefly explain a related topic on learning, hidden non-Markovian signals. So this is a bit more data science, but it's related to the model in the sense that there is another approach that one could take. Could be, instead of fitting the power spectrum, one could take the signal and try to learn with machine learning or any technique the hidden process, which typically would be different to the real process CT. Again, here CT is the center of the trap. So recently, with several collaborators, we put this to the test by doing training algorithms on neural networks. So we presented a neural network a time series and a real signal CT. And we did minimization of mean square loss function in finding what are the most optimal parameters of different architectures of the neural networks. And then we put, after the training, we evaluate how these neural networks work on test trajectories. We typically give 10,000 test trajectories. So our goal is to see if these neural networks work and if they are different, they have different performance. Also because some of these neural networks build an internal variable, which is a sort of memory for this data processing. So in particular, we train, then we produce data and we compute the mismatch error. So how different is CT from the prediction. And very quickly, I can show that in this model that I explained before, there are like two phases. One phase is one in which the XT, so the position of the hair bundle, has a bimodal shape. And another phase is the one in which for example, the switching is so fast that the distribution is unimodal. When you apply this type of techniques, you see that unimodal is much more difficult to learn than bimodal. Somehow, because here there will be a lot of, you will see the particle here or the system here, and you will easily predict that CT is here. In the other case, when it's unimodal, things are tougher. And okay, this was somehow expected, but we tried to compare the error of the learning with a statistical quantity and came up with this interesting parameter. It's called the Sarle coefficient, which is, okay, it's in terms of skewness and kurtosis, it is a measure of bimodality, and we see high correlation between the error of the neural network and how much bimodal is the time series. Instead of the variance, we try also the variance, we try other statistics, but we see that this parameter is really interesting for this and insightful for this problem. This is my last slide with results. So here we compare the different neural networks performance as a function of K. So K is, in our case, K changes the shape of the, I don't know if I can annotate, it changes the shape of the gamma distribution. So when K is 1, it is exponential. And when K gets larger and larger, it gets broader and broader and broader. So the system has more and more and more memory. So we were a bit surprised that increasing memory does not guarantee that the error in learning gets better. At long case, there's a lot of memory and makes things easier for the neural network. But at low case, we are increasing the memory, but yet increasing the error. And we attribute this, we are trying to understand it, to the fact that for low K, the signal X and T are highly correlated. And when we increase the memory in the process, the X of T and C of T become more and more and more independent to each other. So it's much harder to learn here to this point. But then you use a neural network that has more and more memory. So the memory of the neural network beats this effect. Just to, okay, brief announcement is we have a ready-to-use Python toolbox here, which you can use for this task. Okay, another short announcement is there will be an interesting school in Serbia in September. Here are some of the lecturers. Some of them are here in the workshop. Applications are open, so we welcome the very good students and postdocs. Mainly, we welcome students from developing countries, but also students in developed countries which have travel funding are very welcome to apply. So we are not restricted to a set of countries. So please, if you are good students or if you are a good student, think you are a good student, welcome to apply. I want to give many thanks to Gennaro Tucci especially, who did most of this work, and all my collaborators here, and my current group at ISTP. Well, I should say Sarah is now just moved to Cambridge, but I would like to highlight the number of women in the group now in Stochastic Thermodynamics, which I hope is also the direction of the community in promoting women in science. And thank you very much for your attention. I end up with this picture from where we work right now. Thank you.
Thank you for a very interesting talk. Yeah, so I guess we can take a few questions. So I have a quick question. This you showed this ratio of this T effective and T, right? For the one satisfies this fluctuation dissipation and
yes,
one doesn't. So the case where it doesn't satisfy, it seems to go to zero, right? So what does it mean? I mean, this temperature goes to zero, or
not only to zero, it becomes negative. So
yeah. So it becomes also negative, and for large frequency it goes to zero. So what does it physically mean?
The meaning is unclear to me as well as like to you, but here there is a fit to this formula, which is obtained for a very simple model, which is Langevin dynamics, which is linear and has non-reciprocal forces. So you can enforce in that model, even though it's non-equilibrium, because it has non-conservative forces, you can enforce this to be true. In other words, you can compute the ratio between this and this, and you will find that the ratio between these two quantities is not equal to 2 kBt omega. So the wrong assumption is thinking this will work for a non-equilibrium, for a reference process that is non-equilibrium. This works when the reference process, the one in which you compute the correlation function here, is of equilibrium. If not, nothing guarantees that this will take place. And moreover, there is a very good paper of Joanny, Pro, and Parrondo where they generalize the fluctuation dissipation in a reference framework, which is non-equilibrium steady state. I mean, I think he can send you the reference, but it doesn't look like this, so it's much more complicated. And there was a follow-up paper to this one where instead of enforcing the fluctuation dissipation, they look at the generalized one in non-equilibrium steady state.
And there's a question by Parando.
Hi, hello, Edgar. Very nice talk.
Thanks.
So, no, I like very much these minimal Markovian models, but also I think that it is not so bad that the bounds are bad, in the sense that we are only. So I think these bounds are interested not only to estimate entropy, but also a little bit like Mike mentioned this inverse engineering to quantify what is the minimum energy to achieve a given function or to achieve a given behavior. So if you find that with KT you can do what the Eair does with 100 KT, so that's nice. I mean, so in this sense, I like very much the Markov minimal models, because, and I will go a little bit farther. I will say, let's look for the model that reproduces the data with the minimum entropy production. And this is something that Yannick Eric did in a very nice paper. But I think this could be extended. It was for a Markov chain, but I think it would be interesting to extend this, to continue to find what is the model with the minimum entropy production that reproduces my data. What do you think?
Yeah, it's a very good point. And the point is that if you see the oscillations, you could think exactly that there is an optical trap and a colloid. And of course, if you neglect the energy of the laser, so you can do this oscillation with much less dissipation than the Eair bundle. So I think that's a very fair point. Just one thing to add is, you said Markovian, but our model is typically non-Markovian.
Okay, so yes,
but anyway, it's a simple model. And I think what you say is roughly what they tried in one of these papers, in this one. I think they tried to find the two-state model that has the minimal dissipation reproducing two given waiting time distributions.
Okay.
If I remember well, because it's really recent and I have to check in detail. But I think it's a very promising direction, and I agree with you and Mike that
Yannick did this with exploring all the possible Markov chains. And he did that in a nice paper. So it is the same idea. Okay.
But it has also the non-exponential waiting times in his case.
Yeah, I think he was right. He had there was some network, and he looked for all the big networks that could reproduce these dynamics with Markovian dynamics, but with the minimum dissipation. This is in Journal of Statistical Mechanics, I think.
Okay, if you could send a paper in the chat, I'm really happy to take a look and read.
Okay, so if there are no more questions, I have one more question. This since this gamma distribution, I mean the tail is still exponential. Even this Markovian model should explain, I guess, some regime, right?
Yes, true, but we don't know which regime. The first thing, it's a good question.
I mean, large time regime, but I don't know what large time means. I mean,
I'm not sure because if you
look at the smallest behavior of the Laplace transform, I mean, it's the same for both gamma distribution and exponential, right?
But okay, this is long time.
Yes.
And here it appears the Laplace transform of the waiting time distribution.
Yes. So
which is different to the exponential.
No, I'm saying if you expand for small s, it will be like first, then some linear term, which is s, right? And then some higher order terms. So forget about the higher order term. So at least, I guess, I mean, for large time, maybe the tail of W or something, it should reproduce probably, right?
Okay, we didn't think about it, but it could be. I mean, we have in the archive all the appendices, and I mean, it should be easy to find it there. It's I mean, it's very, very long appendix, but it's very detailed. So we didn't think about it, but it's a good suggestion because maybe it helps to calculate some quantities without no Markovianity. Very good point.
So I think if you have a question, Arnab?
One. So this simpler model that it's here in this slide now. Is it relatable to the physiology of the hearing? Means the other model they could easily relate that which parameter is doing what.
Yeah, this is an excellent question, and you have to think that this is a collection of pterocilia that are bending and moving left and right. So somehow this is an effective model. It doesn't have free energies, forces, etc. inbuilt, except the kappa, which is in the end, this is a spring stiffness. But we would like to relate this. So for example, the values, one of the most interesting parameters to me is this K value, which is typically we fit it to an integer number. So when you have no Markovian, this K is greater than 1. And we are trying to relate this number to the number of transduction channels. Because if you think about it, let's say you have the bundle in one side and there are 7 channels. And the 7 channels, they open at random times, like Poissonian random numbers. But you would need the 7 channels to be open in order to go to the other equilibrium position. So if you would build a distribution for 7 units to be open all at once, even thinking that each of the units is an exponential random number, then you will get a gamma with parameter 7. So our a bit intuitive idea is that this number, it's related to the number of transduction channels. And we want to test this in the experiment by, you can have a cell where it has n channels that are functioning, and then you damage 2 of them, and then you see how it oscillates. We would like to fit the oscillation before and after the damage and see if this K is correlating with the number of channels.
With the number of channels. And
please.
yeah, so that the opening time of those channels. This channels are sometimes remain open, sometimes closed. So that thing should, will you think that it should come from this power spectrum of the sea?
Yeah, that's a very good point. And the only problem is the opening time is estimated to be 1 millisecond. So it's really, really fast. And it's hard to see this in the data. But if one could relate this model to opening times and closing times, would be very nice. I don't know yet. We didn't think about it, but maybe the average switching time is related to a mean time of opening of K channels.
Of this K channels.
This could be a very good direction for the future, actually.
Thanks. Thanks a lot.
Okay, so thank you again, Edgar. And I'd also like to thank all the speakers of this session. And yeah, Arnab, it's to you now.
Yes, yes. So we are going to close this session. Next session, we will be there after 1 hour. And we will start the second session of the third day. Okay, so we'll be back soon.